Welcome to MOOC course on Introduction to Proteogenomics. Our next speaker is Dr. Kerstin Krug, who will talk about effect of mutations on signaling pathways and how they could be studied using softwares like MIMP and active driver BD. He will talk about the frequency of phosphorylation and factors which may lead to specific kinase activity. He will also talk about tools like Motif X and Phosphosite Plus for sequence motif analysis and also calculate the frequency of most recurring amino acids near the site of phosphorylation. So, let us now welcome Dr. Kerstin Krug to talk in more detail about the role of various mutations on signaling pathways and also to tell us about various factors which may help us in understanding how phosphorylation can be understood in a biological system.
So I will first going to give you like a short motivation. So why we want to do that, and then I will be very specific, and I will talk about two specific software tools that try to study the impact of mutations on phosphorylation networks. So as Bing just presented, so there's millions of single nucleotide variants known in the human genome, and many of them are associated with certain human diseases, but for most of them we don't know the exact molecular mechanism that causes this genotype to phenotype association. And as we've learned yesterday during Kelly's and David's talk, and also during the hands-on, so mutations can, if they are located in protein encoding regions, they can be non-synonymous, meaning they can lead to a single amino acid substitution, so they can change an amino acid in the protein sequence. And here, so I mean, there's many different synonyms for these events. So non-synonymous SNV is single amino acid variant or single amino acid variant. So these are all refer to the same kind of event, but you will find all of these in the literature. And of course, many of these single nucleotide or non-synonymous single nucleotide variants, they affect sites or amino acids that can be post-translationally modified, like phosphorylation, acetylation, or ubiquitination. And actually, these modifiable amino acids occur very frequently in the human genome. So what you're looking at here are the frequencies of all 20 amino acids in the human proteome. So the most frequent amino acid that occurs is leucine, but then on the second place we already find serine, which can be phosphorylated, right? And what are highlighted here are serines, lysines, threonines, and tyrosines. So lysines are, can be modified by acetylation or ubiquitination. So these are the most well studied, and so we have the technology to study these modifications on a large scale. So that's why I highlighted those amino acids here. And if you just look at the overall frequencies of these four amino acids, they make up for 22% of all amino acids that occur in the human proteome. So it's very likely that a mutation affects these modification sites. And we are asking the question, what kind of consequences does that imply in downstream signaling events? And there has been many studies out there that try to decipher these kind of relationships, and I'm just highlighting a couple of those here. The most basic kind of approach to take here is to look at kinase sequence recognition motifs. So kinase phosphorylates its substrate, and one mechanism to ensure that the kinase specifically identifies its substrate is given by very local interactions. So meaning it's the amino acid sequence, the properties of the amino acid sequence around the phosphorylation site. So basically, as probably many of you have seen these kinds of sequence logo motifs here, where in the center you're looking at the actual modification site. So this aurora kinase B mostly phosphorylates serines, but also threonines. And if you look at the frequency around its substrates, flanking this sequence, you see that there's a strong enrichment of an arginine at position -2 relative to the phosphorylation site. So this is what we call the sequence recognition motif of Aurora kinase B. So basically, it recognizes the arginine at -2. Any questions to that?
So my question is, on what basis we are taking serine threonine in the center, the 0 position?
Well, Aurora kinase, there's two classes of kinases. So one separate class is tyrosine kinases, which only specifically phosphorylate tyrosines. If you look at all known substrate of Aurora kinase, we find most of them have most sites are serine sites, and then there's a smaller fraction of threonine sites. So Aurora kinase cannot phosphorylate tyrosines.
My question was, why is the center at 0?
Well, so we are looking, so this.
We have arginine also. So why we haven't taken that as a 0 position?
So the 0 position is actually the site where the phosphorylation happens. Right? And we are looking around this phosphorylation left and right. Okay. So, and there's many tools that can generate these kind of logos and perform these enrichment tools. And the common principle of these tools is you test for enrichment of amino acid patterns that surround this phosphorylation site. And you compare it against some background data set. So, for example, you have your phosphorylation site data set that you've acquired in your lab. You have, like, in this case, maybe like 20 sites or so. So these can be all detected sites in your experiment. Or if you, let's say, you specifically inhibited the kinase and now you're looking for all phosphorylation sites that are downregulated upon inhibition. So this could tell you these are very likely substrates, either direct or indirect, of this particular kinase. And then you compare the frequencies that you obtained here against the background data set. And this can, again, be all detected phosphosites in your data set, or you could use all known phosphorylation sites in your human proteome, for example. So as I mentioned, there's several tools. So two very popular ones are MotiveX and the sequence motive analysis tool on Phosphocyte Blast. So MotiveX was probably one of the first, if not the first tool, which was published back in 2005 by Steve Gigi's lab at Harvard Medical School. And so the basic principle again, so you have your phosphorylation sites. So the center is where the phosphorylation happened. And then we're looking at the surrounding amino acids. And from that you can very easily build up this kind of frequency matrix where in the columns you have your offset positions. Again, in the like 0 means these are this is the actual phosphorylation site, and then you're looking 7 amino acids to the left and 7 amino acids to the right in this case. Again, this is very arbitrary, so some tools use different, sequence windows length and so on. And on the y-axis, you're looking at like all 20 amino acids. And then you just basically count the frequencies of these amino acids in your data. For example, in this case, we would have like 3 glycines, exactly. We would have 2 prolines, and so on and so forth, right? So it's very easy to build up this kind of frequency matrix. And this is exactly what MotiveX does. So it builds up these 2 matrices. One is derived from your actual phosphosites of interest, and the other matrix is derived from your background data set. And from that, you can then calculate a binomial matrix, a binomial probability matrix, where you, for each position in your sequence window, you ask, and for each amino acid, you ask the question, for example, in this case, how many times do you observe a proline at position +1 in your data set? This you can calculate for each amino acid and each position. And you compare it against the background frequency, which you derive from your background data set. which can again be the entire human proteome or all of your detected sites. And from that you can then calculate or generate these sequence windows. Here I just put up an example which is based on the 7 sequence windows that we've looked at a couple of slides earlier. So if you actually calculate this probability to observe k out of n, so n in this case was 7, so we looked at 7 sequence windows, we observed 4 prolines at position +1, and the background probability of a proline in the human proteome is roughly 0.062, and in R you can just feed it into the binom test function, and you get a p-value that this indeed, although it's a very small sample size, it would be statistically significant. So 4 out of 7, so on and so forth. This again you would do for all amino acids in all locations. So probably don't have to go too much into detail here. So motif X does that like in an iterative manner. So it first of all takes all sites that you feed in and extracts the most significant sequence motifs from that set, and then it removes those from the initial set and repeats the analysis. So that's one specific property of the software. Okay, so now we know how we can look or how we can determine these specific kinase sequence recognition motifs, but now what happens if if a mutation happens in this kind of region around the phosphorylation site? So there's actually 3 different scenarios that can happen. So one is a direct hit. So this is the wild type here, this is the mutated version. So in the wild type you have a serine which is actually phosphorylated, and this serine now, due to a mutation, gets mutated into a histidine. So it cannot be phosphorylated anymore. And of course this can also happen the other way around, right? So the histidine can be mutated into a serine, which now maybe present a new phosphorylation site which can be recognized by a kinase. So it can be either it can lead to a genesis of a phosphorylation site or to a destruction of phosphorylation site. So the other possibility that could happen, so it does not happen at the exact site, but it can happen very close to the phosphorylation site. So in this case we have this proline here at +1, which now is mutated into arginine, and the kinase that was able to recognize this proline can now not phosphorylate this specific serine anymore because the proline is gone, right? So meaning in this case we would lose this phosphorylation site. Again, it can also go the other way around. Or it can also happen that you just change the sequence kinase motif. So in this case, in the wild type, it was kinase A who recognized this motif, and now due to a mutation, this motif changed into another motif that can be recognized by kinase B. So these are the most simplest examples of these kinds of events that we are looking at. There's also further events that happen further apart from the phosphorylation site. For example, if a mutation hits a kinase domain, which contains the catalytic function of this kinase, it can also change it can lead to aberrant kinase activity. So here on this slide, I just presented a couple of tools. So there's many tools out there already, and people have started looking into that 10 years back already. But also recently there are lots of new developments. So these are actually the 2 tools we are going to have a closer look at. So one tool is called MIMP and the other is called Active DriverDB. Just want to highlight, so this other tool here, G2P database, so genome to phospho database, which has been developed in David Fenio's lab as well.
So in Motifex, I tried to use that tool. So I encountered one problem here. I have a phospho site and the residue phosphorylated. Like the serine is phosphorylated at 32, and I have the sequence, peptide sequence.
Yeah.
But there it was asking which site is phosphorylated. Like in the peptide, might be that the serine position is 4th position.
Yes.
But in my sequence, it was serine because I am a biologist, I am not a programmer.
Yes.
So is any one of these tools, as you know all of these tools, is any one of these tools solve my problem?
No, well, these tools don't solve your problem. So this is one step actually upstream of this type of analysis. So many of these tools that actually take the raw mass spectrometry data and do the database search and create result reports on a phosphocyte level, let's say. So many of these tools already have these kind of sequence windows in their result table. So this is actually not a peptide sequence, right? It's the sequence window which is always the same length. So in this case, it's always 15 amino acids. It's this, and the modification site is in the center. So this is something that many tools create, like MaxQuant does it, like SpectroMill does it. I'm not sure whether Proteome Discoverer does it as well.
In Proteome Discoverer, I got peptide sequence and
Yes.
it says serine 32.
Yes.
And might be my serine is at the 4th position. So
Yeah.
I was not able to use
Yes.
as I said, MIMP tool and this modifiers.
I see your problem. So what you would have to do, you need to hire a programmer that just takes the data, takes the database and creates the sequence windows. So, or you just use another software, so I can highly recommend MaxQuant. Okay, so MIMP does exactly what we've just talked about. So it predicts the impact of non-synonymous SNVs on kinase substrate interactions. So it predicts kinase binding affinities and how or whether a mutation rewires protein or like phosphosignaling networks. And it compares, so that's basically like the principle here. So it compares the effect of mutated and wild type samples, and one specific property of this tool is that it uses a Bayesian approach to construct like these, in this case, position weight matrices, which is very similar to these amino acid frequency matrix that we just looked at. So this has been published in Nature Method like 3 years back, and there's an online tool which you can just use, but there's also like an R implementation of that package, and this is actually what we are trying to use in the hands-on session. So I hope that we will be able to do that. So we'll see. So the key features is it first builds kinase binding models using known substrate sites. So very highly curated, very well-known phosphorylation sites that have been determined to be a substrate of this particular kinase. It used this information to build binding models. And these models are already included in the software, right? So this is nothing that you have to worry about. So then calculates for a given phosphor sequence that might be now coming from your data set, it calculates the kinase binding score. For each kinase, it calculates the score, how likely it is that this phosphosite has been phosphorylated by this specific kinase. And again, so you can upload your own phosphor sequences, or you can just query all phosphosite sequences in Phosphosite Plus. Does everybody know about this resource here, Phosphosite Plus? So I can, again, highly recommend to check that out. So at the end of my slides, there are like all references that I'm going through here are included, so you can just go to the papers and check them out. Yeah, so the first part is calculating kinase binding specificities, and then it pulls in mutation data, which again you can upload your own mutations, or you can specifically look for TCGA mutations, and then it does its prediction.
So today, in conclusion, I hope you have learned that kinase activity gets affected due to the amino acids which are present in the surrounding of the phosphorylation site. Hence, if we know the correlation of amino acid to phosphorylation specificity, then we may change the expression pattern of a gene. We also heard that motif X follows an iterative workflow which provides us reliable and confident amino acid sequence which could result for the phosphorylation regulation. Mutations can lead to genesis, modification or destruction of P sites resulting in altered pathways. Dr. Krug also enlisted many tools which can be used for correlation between genomic mutations and signaling pathways. The next lecture will be the continuation of mutation and signaling by Dr. Krug. Thank you.